Organisms, we talked about them. These are multicellular organisms, metazoa and parazoa. I will give you a definition of each one of them in a minute. But here they are. There are three possibilities that metazoa means multicellular organisms. That's what metazoa means. And parazoans mean they evolve parallel to metazoan. Okay, they are parallel, but they are different. That's what parallel means, parallel to metazoan. And then they came from syncytial ciliated cells. Syncytial, it means a cell that has multi-nuclei. Can you give me an example of a syncytial cell?
Skeletal muscle.
Yeah, a skeletal muscle. Ciliated, some of cardiac muscles. Ciliated. Paramecium had nuclei, I should say, two nuclei. So syncytial ciliated, that's one theory. Okay, the second theory, clonioflagellated. Can you give me an example of a clonioflagellated organism you studied already? Volvox. Volvox, as I said before, somebody asked you during review day that why is it important? Volvox is important because multicellular organisms, they came from something similar to Volvox. Okay, so and then finally, monophyletic, it means one phylum, monophyletic and not polyphyletic multicellular organisms. I'll put this because your textbook changes from one edition to the next edition. You have the older edition, it probably says polyphyletic. Polyphyletic, it means multi-phylum. This is one phylum. They evolved possibly from one phylum, and then organisms evolve from one phylum and not from multiple phylum. And then based on ribosomal RNA in the lab, you saw the video and said, what is taxonomy? Classification of organisms based on, classification is pretty much based on DNA and the morphology, homology, similarities between the organisms. That's how they put them in different taxa. 100 years ago, they didn't do that. They didn't use DNA. They just used, if you have two arms and another animal organism has two arms, you put them in the same phylum, for example. Okay, they used homology. They didn't use analogy, for example. And still, in the past, they didn't use it. Now they don't use it. Analogy is, for example, bats fly, right? And birds fly. So let's put them in one phylum. No, that's not how it goes. The structure, the homology in the bat's wings is different than the wings of fly. So they go to two different class in this case. Okay, but anyhow, but now in order to figure out this a little bit more, not only they went to the nuclear DNA, they tap into the ribosomal RNA to get into similarities, because the DNA could be very similar between two species. So what they did, they said, okay, let's look at something else that could be a little bit different, than that would be ribosomal RNA. And from biology 1, what are different type of ribosomes? Anybody? We have 3 different type of them. What is it? But not ribosome, I'm sorry. There are 3 different type of RNA. Sorry about that. I'm confused.
Messenger transfer.
Very good. Messenger RNA, is that right? rRNA, ribosomal RNA, and tRNA, transfer RNA. So they tap into ribosomal RNA, they get ribosome, and they get RNA from it. So, and they make analogies, what organisms came up, and they try to prove it using rRNA. Okay, there are 5 grades. Now we're going to get into the area of grades and clades and so on and so forth. Yeah, you should know some because it might come in handy later on in your life. In this class, sure, yeah, but it depends what classes you will take later on in your life. So it might come in handy. The first grade is a grade protoplasmic grade, like paramecium. An example of that would be paramecium organisms, amoeba. Those are all belong to the grade protoplasmic. So you have the kingdom, then you have the grade, then you have a clade. All of those has it's different than what you learn in biology. Well, in biology one they talk about kingdom, but it's a little bit different than what your old edition of textbook is talking about. Then the next one is cellular grade, like ball blocks. Okay, I should have underlined these organisms, ball blocks. And then cell tissue grade, which that is this exam material, jellyfish, hydra. You don't know what hydra is in a minute, not in a minute, next week. You will know hydra like back of your hand. It's a very simple animal. And there's not much into it. So it's same as jellyfish. And then we have tissue organ grade, which is flatworms, planaria. Again, these are going to be in this exam, so all of this is going to be in this exam, tissue organ grade. And then next exam material, which is organ system grade, human, earthworm, all of the organisms after flatworms, they are pretty much in the organ system grade. Okay, I hope you know what I'm talking about when I say grade and clade. It will come up, all of that will come up. Development of patterns, protostome development, 2 clades or group are and more are in this Lophotrochozoan. This is a clade. Within a grade there are clades. So, and within clades there are phyla. Okay, I hope I'm breaking it down for you a little bit. And then segmented worms, mollusks, and others. So that would be an example of Lophotrochozoan clade. Have horsehair shape, whorl of tentacles called lophophore and trochophore larva stages. Those are some of the characteristics of the clade Lophotrochozoans. The other clade is ecdysozoan. Ecdysozoan is the arthropods, roundworms, and some other groups. I don't know, some of you probably are familiar with this term, ecdysis, molting. How many of you have heard of the term molting? Animals that shed their exterior layer, their outside layer, they shed it, they release it, okay, and the new one grows back. Okay, that's called ecdysis. Okay, molting. So animals like roundworms, which we will study in exam number 3, arthropods, which are your flies, crab, cockroach, they all are arthropods. They also shed their exterior layer. So it's called molting or ecdysis. And they belong to clade ecdysozoans. Yes?
Would like snake belong to ecdysozoans?
No, they put snake in our clade, which is the next one, able to, snakes does not belong to that group. It's, yeah, they don't.
Okay.
I don't want to, it's not, the snakes is more complicated. Just like us, they shed the whole entire skin, but we shed our skin too.
Oh.
Right?
Yeah.
So it's different type of molting.
Yeah.
The reason why I say that because of the enzymes that are involved in molting.
Yeah.
In us, they just grow and they die.
Yeah.
Okay, that's pretty much what it is. Okay, I just talked about 2 clades. There is more, but anyhow. Here it is. Pretty much, I'm not going to read it, but this is in your textbook. It has examples of the 5 clades. 5 grades. Sorry, I'm getting them mixed up now. While I'm up here. So there are 5 grades that they are mentioned in your textbook, and here are the picture. Okay, taxonomy. Still, we use taxonomy is classification of organisms. They use them in botany. They use them in microbiology. They use them in mycology, study of fungi. That's for everything. I mean, classification. It's the bottom rock of everything. It's just like they ask you, where do you live? I live in Stockton, right? Where? Stockton, where is Stockton? You go, for example, you go to Japan, and they ask you, where do you live? I live in Stockton. You say, where is that? Oh, it's in California. Where is California? Oh, you don't know where California is? No, I don't know where California is. It's in the United States. Where is the United States? Oh, it's in North America. Ah, I know where North America is. You see what I'm saying? It's a way of communicating between people. That's what taxonomy is. It's a communication thing. And I hope I made sense. I did talk about it a little bit. So, but anyhow, these are the clades. Again, it depends who you grab collar of and talk to them. You grab my collar in my collar in the mall and say, Amir, what are the clades? What are the kingdom? What are the grades? I said, I don't know, but these everybody agrees on. The seven mandatory majors ranks King Paul came over for good 6. Remember that? We talked about this. Okay, all animals are placed in Kingdom Animalia, for sure. Each major rank can be further studied, subdivided, and so on and so forth. Superclass of order, blah, blah, blah. And more than 30 taxonomic ranks are now recognized. That's a lot. During my time, it was probably maybe 10, 5, I don't know how many, but now it's 30. And there are 34 different animal phyla, but you don't have to memorize these numbers. I never ask you how many different animal phyla are there. Roughly about 34. Okay. Major subdivisions of animal kingdom. Phylum is the largest formal taxonomic category in the Linnean classification. Eometazoa includes all animal phyla except Porifera and Plecozoa. And I will go over Porifera today and Plecozoa. The rest of the animal in animal kingdom are eometazoa or metazoan. Eo at the beginning of a word, it means true. Okay, true animals. But so they call them eometazoa or metazoa. And then major groups are radiata, which is Cnidarian, and Ctenophora, and bilateral are all animal groups. And we'll get to these two, and I will talk about it a little bit later on. Bilateria is further divided into protostome and durostome animals, which you're already familiar with that. And then the Mesozoa, which I only have maybe one slide or two slides, still a controversial group with Eometazoa. So Mesozoa, still some scientists would like to argue about them. But anyhow, phylum, that's what Mesozoa is. Phylum, Mesozoa. They are very small animals, very small. They are ciliated, they are worm-like, wormivore. And then they're all parasitic of invertebrates, example octopus, squid. I have to say octopus twice. Octopus, right? If I have a,
it's just that important.
If I have a third child, I'll probably call him or her octopus. Some people do that. You guys don't remember the United Nations. You all know, who does not know what the United Nation is? You all know what the United Nation is. The chief commander, the top-notch person of United Nations, he was from Egypt. You guys look it up. I'm not lying. I'm being recorded. His name was Boutros. His parents must have liked the name Boutros very much. Boutros Ghali. That was his last name. Boutros. So I don't know. You never know. If I have a child, I'll call him Amir. Instead of just one Amir, I'll call him Amir. I don't know what happened here. Some of you, Nicholas, sabotaged my PowerPoint. So, but anyhow, adults called vermivore. As I said, they are vermivore. Term, you should be familiar with that term. Vermivore means worm-like, like a worm. Okay. Here they are, some pictures of Mesozoa. I thought I put it in there so you have some pictures that matches. Let's go to Plecozoa, glides over their food using digestive enzymes, and that's all I have of Plecozoa. There's not much, I don't even have a picture, nothing else I have to say about them. Then the Porifera, which you saw a video of it this morning in the lab, let's elaborate on them and talk about them. And then on today's Thursday? Tuesday. Good. On Tuesday, I don't know what day of the week it is. I just get up and go. You guys. And then during the exam or quiz, you ask me, what is the date today? I said, I have no clue what day of the week it is. You ask me the date.
Do you have a month?
Yes.
Is it vermiform and worm-like?
Worm-like, yes. The term vermiform with V at the beginning, vermiform, it means worm-like. Yes. Okay, phylum Porifera. So quiz on Thursday, Tuesday next week. It's over all of these slides. Wherever I stop, I think I will finish up. Hopefully, we'll finish up sponges today. I hope. Okay, you saw the video on sponges today, so everything on the video is also a fair game. Sponges are simplest animals on planet Earth. Yeah, until what? 200 years ago, 100 years, well, not 100, 200 years ago or so, 250, they thought sponges are plants because they don't move. They're stationary. And they thought they were plants. But now, after studying them more, they realize they are not plants. Filter feeding system by Osteo, and I'll go over that today in a minute. Hang on, give me a rain check on that one. I will talk about it. Sessile, and they're all aquatic. We do not have a terrestrial sponge. All of the sponges are aquatic. The one that you have it in your bathtub, you use it to clean yourself, it was aquatic. It was in the ocean sometime, somewhere. And they harvested on the turn of the century, 1900, they stopped harvesting sponges because they were going to be extinct. They harvest them for cleaning dishes, cleaning the bath, everything. Now they're a little bit more expensive, and now we have synthetic sponges as well. So now their population is coming back. The bath sponges, I'm pretty much talking about bath sponges. So here they are, spicules made up of calcium carbonate, and I will talk about spicules. I'll give you a picture here in a minute, hang on. Silicon or spongin. Spongin is a type of spicules that is made up of collagen. And you're familiar with the term collagen. Collagen, as I said before, is the most occurring protein molecule on planet Earth. Imagine just in this room how much collagen we have, whole bunch, under your skin. Okay, so spongin is, we have slide of spongin in the lab. You should look at it. We have a slide of spicules, the calcium carbonate or silicon spicules in the lab. Asexual and sexual reproduction sponges are microorganisms living on them often have a noxious odor and produce a variety of bioactive compounds. Certain sponges extracts have manifested medical and pharmaceutical effectiveness. I thought I'd put that in. It's in your textbook, some of that information. I thought I'd put it in there. If somebody look at the sponges PowerPoints, and then these are not in your docu-share, let me know, so I put these up, okay? So they have a nauseous odor. Just recently they found out they have some of them. Okay, here are the spicules. You see how sharp they are? And this is a spongin. It looks like spaghetti. You do have a slide of this in the lab. Okay, so it looks like spaghetti. It's made up of collagen, but this is made up of calcium carbonate. This is made up of silicon, and they're all shaped. Sponges are classified. When I say classified, you all know what I'm talking about. Class, order, genus, species, based on, based on what? I just want to see you're alive. I know you're all sitting in there, but I just want to see you're alive. And the school does not allow me to go and touch you guys if you're alive. So I just want to see what. So the classification of sponges is based on the spicule and also the shape. So, and of course DNA comes into picture for everything from now on for the rest of your life. But DNA, shape, and spicules. Okay, Ruben, are you farthest away from me? I cannot come and touch you.
No, I don't mind.
No, you never mind. Okay, here they are. I have a general picture of filter feeding, what happens in sponges. Okay, so this is the story. If somebody took my ruler, well, I'm glad nobody's sitting here anymore. Wesley, you remember first day? Half of you guys were sitting on that end. I said, oh God, what am I supposed to do? What if they don't decide, oh, come on this side? I said, if one of them, if Ruben sits there, what am I supposed to do? I must constantly call his name. Ruben, pay attention. It rhymes, it's a rap song. Ruben, pay attention. Okay, so what happens, this is the sponge sitting on the bottom of the ocean or lake. There are some species that are found, a few species found in the lake, but in fresh water. When I say lake, fresh water. Okay, so this is outside, the water goes in, and then that water comes out. That's what filter, and then you have the wall of the sponge here, that inside of it is hollow, inside of sponge is hollow, and we call it sponge seal. And then right here, when the water is going from outside, you see the arrows, the flow of the water. The water goes from this outside, these openings are called ostium, singular, ostia, plural, just like everything else in biology, plural and singular. Okay, then you go out through osculum. Okay, so ostia, plural, and ostium, singular. And then what happens, these little specks, you guys see that? Those little specks get absorbed by these choanocytes and the flagella. They have flagellum, I should say, and they push the water out, and they have microvilli, which absorbs the food and nutrient. And that's pretty much what filter feeding is. There's a little bit more detail, but I just gave you a big picture, and then we come back and fill in all of the details. Aubrey.
Are those orange things spicules?
Yes, thank you so much. And these orange things are spicules, which the function of it in the lab you learned today is rigidity, support, right? It gives me support. Am I making some sense? A little bit. I'm supporting myself. How? Besides delta knowledge is supporting me, but besides delta knowledge, my skeleton, right? My bones, that's supporting me. Same as these guys. They are supported by these rigid spicules. Okay, how are we good? Should I go? Okay, 3 types of canals, yes, and when you will see pictures of them. Ascenoid, flagellated sponges, they're simplest type of canals. Out of the 3 type of canals, that's what I mean, they are the simplest one. Leucosolinia is the name of the species, which you do have in the lab. Maria, yes, am I making some sense? Yeah. The second one, cycanoid type canal. The cycanoid type canal, I enlarged it because we have it in the lab. The slide's microscopic slide of cycon. The name of the, I underline it because of what? It's the name of a genus. I underline it because it's the name of a genus. Okay, so cycanoid, flagellated canal, larger edition of ascenoid, and you will see a picture of it. If I go back, the last slide, you can go back on YouTube. That was an ascenoid type canal, and you will see it again. It's going to come up. And the final, the most complicated of the 3 canals, the most complicated one is leuconoid canal. Flagellated chamber, water current is through, and I will show you some pictures. An example of that would be bath sponges. I do not have the name of a genus for you. Okay, just regular bath sponges in the lab, you see, I might ask you what type of canal system that is. That is your leuconoid type canal. I mean, the names are kind of similar to each other. I'm sorry about that. I didn't make up the names. If it was up to me, I would call everything Amir. Just for simplicity reasons. Just for simplicity. It makes everything simple. Why are you guys making things difficult?
All taxonomic classifications. Mirror.
Types of cells found in mesoheel, and I show you guys a picture. The mesoheel, mesenchyme, mesoglia, 3 names for one thing. That's another crazy thing about biologists. Not only they don't call everything Amir, they call one thing, they call it 3 different names. Why? Why do you do that? You like to confuse students. Yeah, that's what they do. But anyway. Pinacocytes, they help to regulate, they are on the surface of the sponges, and they help regulate the cell, and they are epithelial-like cells. And you all know by now what is epithelial cells like. Okay, you have 3 different, I don't know, in the past, but most these look like squamous epithelial, depends on the species, but you know. Choanocytes, straining food particles. So portion of choanocytes is in mesoglial layer, and portion of pinacocytes, you will see it, portion of it, not all of it, is also in the mesoglial layer, say mesoheel, mesenchyme layer. And then finally, all of archaeocytes are in mesenchyme layer. And you will see a picture of it in a minute. Hang on. Easy. Okay. Archaeocytes. Archaeocytes, they're amoebocytes, they're amoeba-like cells. And phagocytize is the food that comes, that is the animal's filter. Remember filter feeding? I talked about it. The animals do filter feeding. It helps you. I asked you guys to look at the PowerPoints or watch the videos before you come to the lecture. And now if you come to the lecture, your brain is more like a sponge. Okay, but if you didn't, that's okay. I mean, you have to hang in there, but no daydreaming allowed. I should put it on every slide. I should put a sign right here. No daydreaming allowed. Right? Yadi, Jasmine, you guys are with me. Okay, here they are. There are 3 different types of canal system. Let's go over them quickly. The first one is the most simplest one, ascanoid type canal. An organism like that is leuconoid. We do have them on the microscope, small animal. We do have them on the microscope. So what happens, water comes through ostium. These are ostium. It comes in, and then there is food in here, and food gets trapped by microvilli of the choanocytes. This is choanocytes, right? These cells right here are called choanocytes. And then choanocytes have these little flagellum, and the flagellum push the water that has been filtered out through ostium. Very simple type of canal system. Okay, this is the one you do have slide in the lab, is the cycanoid canal system, and the name of the organism in the lab, they italicize it, is cycon, okay, or grantia, or there's another name. There are 3 names for the same species: grantia, cycon, or anyhow, I can't remember the other one. So here is the opening right here, is called ostium. So water comes through ostium and goes through these canals, which is called incurrent canal. Right here. It's called incurrent canal. So the water goes through ostium to incurrent canal. From incurrent canal, water goes through these opening, which is called prosopile. It's not mentioned in here, but here, right here. It's mentioned right here, prosopile. So water goes in from prosopile, goes to radial canal. These are radial canal. And then inside of radial canal, you have these choanocytes. All of these choanocytes that I mentioned, they're right here. And they absorb food. And then with the help of the flagellum, they push the water out, and the water goes out through osculum. And that space right here, look at that space. That space right here is called spongocele. Seal on your prefix suffixes means what? Yeah, space right here. You have space. Spongocele. So they picked the right name for it. Seal, space. So that space is called spongocele. And they push the water out here through the choanocytes are lined up in spongocele. Here, choanocytes are lined up in radial canal. I hope I'm making some sense. And of course, the opening between radial canal and spongocele is called apophyte. Soon, Christmas or Thanksgiving is going to be here soon. Before you blink your eyes, that's how fast time goes in this country. Other countries, I go, it's more relaxed. Time goes slower. But in the United States, I don't know why time goes so fast. If you blink your eye, it's going to be Thanksgiving, and you're going to be eating. You guys don't eat apple pie. Do you guys eat apple pie?
Thanksgiving?
Thanksgiving, yeah.
Pumpkin pie.
Pumpkin for Halloween, isn't it? For Christmas. Forgive me. I'm sorry. For Christmas. Whatever. So here, so that's how that I ask you guys, what is the movement of water in sponges? Hopefully, I'm in cycanoid type canal. I just said it. Right here is the movement of water. And then how does filter feeding occur? Then you should explain movement of water, and then food is absorbed through the choanocytes. Okay? So, and I'll show you a bigger picture of it, of the choanocytes, how they are absorbing food and nutrient, and they give them to amoebocytes, right here, archaeocytes. Archaeocytes are right here, archaeocytes are right here, and those archaeocytes absorb the food that comes from choanocytes, and they make up spicules. They do all of the goodies they're supposed to. Okay, so the last one, which we do not have slide of it, and I will not ask too much, maybe I guess you should know it. It's a leukonoid type canal. Leukonoid type canal, they have a chamber full of the choanocytes, and then same thing happens. Water comes in, and then goes through these chambers, and then it goes out through excroin canal and osculum on the top. So that's what happens in the leukonoid type canal. But this is the main thing I would like you to know. We do have slide of this. We do not have slide of this. We do have slide of leukonoid, but it does not show you the canal system. Okay, this it shows you they cut it. They cut the sponge longitudinally, so you can see. They cut the sponge longitudinally, so you can see all of those structures in there, and you do see them on your microscopic slides. Okay, is there any question before I move? Yes.
What did you say the name of the cells that create spicules?
The what name of what?
The cells that create spicules.
Okay, archaeocytes or amoebocytes, and I will talk about it. Archae, it means ancient. Archaeocytes, site, it means cell. Or another name for them is amoebocytes. Some textbooks like to refer to them as amoebocytes. Your textbooks refer to them as both. Amoebocytes and archaeocytes. Yes, sir.
So the osculum is basically the ending of the sponge's.
Very good. Osculum is on top right there. Osculum is right top there. Osculum is right top right there. Yes. Okay. Here we go again. This is the cross section, if you would, of the asconoid, cyclonoid type canal. And right here is the apopyle. Here is your radial canal. Here is your incurrent canal. Here is the prosopyle, and so on and so forth. You can see them all. And here, that's what I was waiting for. The choanocytes. We do not have slide of this individually in the lab, you guys. They have a nucleus, and they have these microvilli. Did they mention it anywhere in here? These are called microvilli. Where else have you seen the term microvilli? Right?
Intestines.
No. Very good. In your small intestine, those simple columnar epithelial tissue, you remember how that information is coming with you? If you didn't study them, you didn't know them, I don't know, you better learn them now. So right here is the brush border, the microvilli. So they're extension of the cell membrane. So these food absorbed through here, and they pass it on to amoebocytes here. Right? You all know what I'm talking about. And then of course, whatever is left, these flagella are beating, and they push the water out through their spongiocele, and then out the apopyle, spongiocele, osculum. It depends what species we're talking about. Here they are, reproduction. We do have this in the lab, gemmules. Do not worry about all of these structures. I'm not too crazy about these structures, but we do have gemmules in the lab. Asexual and by budding and regeneration, gemmules, internal bud, that's what they are. You have them tolerate harsh environment. When parents die, gemmules survive. Sexual reproduction also occur. They're monoecious. The term monoecious, you learned it somewhere in your life, high school, bio 1. And what does that mean? And monoecious means very good. One what? Very good. You got it. One species that have both sexes. Have you your classmates or your teammates in the team, you're hermaphrodite, it means you, yeah, no, you don't. In college they bothered me. Nowadays they call them bullying, is that right? They did to me all the time. And I was a freshman, sophomore, junior, I can't remember what year I was. My friends used to call me, hey, hermaphrodite. I said, yeah, you are too. So, but anyhow, that's another name for monoecious. Monoecious, it means when both sexes. It means that the reproductive tract of male and female is in one organism. In plants it's common, but in animals It happens. I wish I could say how many percent or what, but it happens right here. It's one. Most of the studies, organism we study at the until next exam, most of them you will see they are monoecious, but after soon you will see that they are male and female. Sperm and oocysts, you're familiar with that term, are formed from choanocytes and archaeocytes or archaeocytes. So sperm and oocysts. Oocysts, it means the reproductive cells, the eggs in this case. And then viviparous releases ciliated and larval oviparous external fertilization. I might as well say that those three terms, two of them I mentioned in here. So you have, I will give you the definitions. You have oviparous, and then you have the third one is, let's go over them right now, so for the rest of the semester I'm free. It's called ovoviviparous. Ovoviviparous, well, let's go over it. Viviparous, right? It means animals, the whole entire animal kingdom. Animals that give birth. Animals like human that gives birth are viviparous. Animals that give birth are viviparous. Oviparous, it means animals that lay egg. They lay egg, and the egg stays in the environment for a while, 2 weeks, 3 weeks, depends on the species, a week, and then they hatch. Like birds. Birds in general, they're a good example of oviparous. And then we have animals like shark, ovoviviparous, like scorpions, ovoviviparous. These animals, which sponges do not have them. Sponges cannot be ovoviviparous. Sponges are viviparous or oviparous. Animals that are ovoviviparous, like shark, scorpions, they lay eggs, but the eggs hatch immediately. It does not have to stay in the environment for a long time. When you look, if you saw some videos of shark giving birth, I say giving birth, but they do actually have eggs. The eggs are fertilized, and then they come out of the oviduct. Okay, scorpions, they lay eggs, and immediately those juveniles go in the back of the mother scorpions. I don't know if you've seen pictures on YouTube. Okay, so those are examples of ovoviviparous. They lay eggs, but the eggs immediately hatch. An hour, day, time. Okay.